Hi everyone, I'm Baihan Lin from Columbia University, and today I will present our paper, Single-Cell Topological Simplicial Analysis Reveals Higher-Order Cellular Complexity. Here's our agenda for today. I will first motivate the talk with background, explain our methodology, and then go into our results, followed by a conclusion. So background, what do we care about? The single-cell data are very noisy, and one way to better analyze this data is to use topological data analysis, which is a set of unsupervised learning methods that emphasize the manifold or shape of the data points that are invariant to the noise level. scTSA is earlier work that proposed to use topological data analysis as a prior to select significant genes in single-cell RNA sequencing and resolve asynchrony and continuity in cellular identity. So let's consider the following single-cell RNA sequencing dataset of the vertebrate embryogenesis. We see that it is a large dataset with over 38,000 cells across 25 cell types and collected from 12 different time steps. If we select the top 103 genes from the scTSA pipeline and then apply the mapper algorithm to map out the graph structure of the dataset, we get the following plot, colored by the timestamp the sample is collected from. We quickly notice that the 12 time points are not perfectly mapped here. We see different time steps mingle together, which makes it hard to extract the temporal sequence of gene expression profiles. This is complicated by the fact that, unlike the dataset used in the scTSA paper, not all gene expression profiles can have a clear temporal structure that is a skeleton or flare. So a question we aim to answer in this work is that, are there more distinguishable time-dependent features we can extract, other than the relative distance in TDA? Can we characterize the time point better? Our work aims to solve this by proposing new ways to characterize the temporal skeleton and higher-order summary statistics to categorize the geometric invariance beyond what people usually compute, which is the Betty number after order 1. To tackle the second point, we seek insights from the simplicial architecture of the data points. A simplex is a geometric object that is formed by a combination of the same geometric object in a lower order. For instance, a zero simplex would be just one point or cluster, and one first simplex would be a line or edge that connects 2 points, or two zero simplices. By going into higher order objects, we can have cases where an n simplex is formed by several n-1 simplex connecting to another under certain rules. In this work, we use the Vittorius-Rips complex, which is an easy way to compute the simplices by forming a simplex if its lower order simplices are all within a distance lower than the threshold epsilon. Back to the scTSA paper, a concept of cell complexity was defined as the number of genes whose expression is detected in a cell. But the correlation between the sampling time points and the cell complexity are not obvious. We will propose a new definition of cellular complexity. Here, we are interested in the intercellular interaction within the same type of data, same type of cells. In this case, within each time point, rather than their relationships as in scTSA. For instance, do the cell has a homogeneity that makes them not solitary but collaborative to one another? Or are they all similar to just one another cell as a chain, or all similar to one single cell as the protocells? As a teaser, our approach involves witnessing sampling of large amount of data, computing the Rips complexes, and then comparing their simplicial architectures. Here's our scTSA framework, as in single-cell topological simplicial analysis. We first pre-process the single-cell RNA sequencing data, and then compute the persistent homology based on the data sample from all the cells. During the filtration process, a simplex tree is created that contains simplicial complexes of different orders. The number of these nth order simplicial complexes are used to compute our topological invariant summary statistics. After this step, we further analyze the higher dimensional simplicial architecture directly, treating them as summary statistics, or perform temporal filtration for time series data, and then visualize them with the mapper algorithm. Here is an intuitive example of our proposed temporal filtration. Shown here are 7 data points marked with their time steps. Traditionally, we only consider their spatial relationship. As in the leftmost picture, we will see a loop. However, if we add in a temporal constraint tau, it can be extracted out more temporally sequential skeleton. For instance, if we limit the edge forming to only the pairs that are temporally adjacent, as in the second point, we have 3 disjoint groups. By changing the temporal constraint, we can see different temporal skeleton. In the persistent homology, we perform the filtration from 0 to maximum distances, and then mark the birth and depth of this emergent topological object. An equivalent visualization of the persistent homology is persistent diagram, which marks its barcodes into points onto a 2D space. Over the filtration process, we count the number of simplicial complexes of order from 0 to 1 to 7. The normalized cellular complexity is then computed by taking the ratio between the number of simplicial complexes in a certain order n computed from the actual data, and those computed from the control model. The overall pipeline of the temporal topological data analysis is the following. First, we subsample the timestamps high throughput data points. Then, we select the top features after certain dimension reduction techniques, and then either split them by the timestamps, or perform temporal simplicial analysis, or applying their temporal proximity as a constraint for downstream temporal persistent homology computation, or temporal mapper or visualization. Again, here is an overview of our single-cell dataset after pre-processing and applying different dimension reduction techniques. Shown here are the temporal and non-temporal persistent diagrams, where we didn't observe a clear difference. When we expect the simplicial complex and normalized simplicial complex, in summary, these two figures both suggest that the gastrulation stage, time points 5 to 6, is a very critical stage in vertebrate development, matching the established understanding in the developmental biology that it is a process where embryo begins the differentiation process to develop into different cell lineages. Before gastrulation, the embryo is a continuous epithelial sheet of of cells. After the gastrulation stage, organogenesis starts to be where the individual organs develop within the newly formed germ layers. This observation is further supported by the visualization of topological data analysis mapping. Here, we compare the network visualization with or without the temporal filtration. Temporal filtration identifies the critical stage of cellular complexity changes. The color indicates time points, and each node corresponds to a small cluster of cells or collected at the same time points. Conventional TDA mapping identifies the bifurcation structure, but there are spatial locations that has a mixture of clusters that belong to non-consecutive time points. This makes identification of the developmental pathway challenging. When applying the temporal filtration, like the right panel, the mapping identifies a cleaner separation of two tracks or two subpopulations of cells that evolve in the gastrulation state, matching the observation in our summary statistics from the algebraic topology. In order to identify and investigate the trade-off between the higher order and lower order simplicial complexities in the developmental stages, we map the normalized third order simplicial complex against the normalized first order simplicial complexity. Our results suggest a gradual increase in higher order complexity starting from the fifth time point, and overall or below null lower order complexity in a monotonically increasing direction since the second time point. Comparing to a null model, the presence of a much larger number of cliques across a wide range of dimensions in the single cell data suggests that the connectivity between the cells might be highly organized into numerous fundamental building blocks, as for instance some proto data types with increasing complexity. This filtrated simplicial architecture may also offer insights in cell lineage tracing. We performed the hierarchical clustering of the summary statistics computed from the transcriptome data of different cell types. We compared the results using the proposed normalized simplicial complexity versus the one using Betty numbers, which is more conventionally used as in many downstream topological data analysis. As shown here, the normalized simplicial complexity offers a more reasonable clustering performance as a more distinctive summary statistics than the Betty numbers by itself. In summary, our work describes a novel, scalable, and unsupervised machine learning method that facilitates the understanding of and solutions to three main technical challenges in bioinformatics. The topological features identified from the pre-process can be further applied to downstream ML tasks, such as hierarchical clustering of cellular lineages. First, usually we have a time series analytical method in quantifying underlying temporal skeleton within the manifold of the similarities among data points. Second, we have a scalable computational method for characterizing single-cell sequences signals in a scale of over 10,000 data points. Third, we can have insights and interpretation that connects the mathematical languages of algebraic topology to the physical references to biological phenomena. In addition, we propose a new family of filtration or for longitudinal time series, multi-dimensional data, along with auxiliary data analysis tools. We demonstrated our application to the temporal inference problem using a set of time-resolved gene-expressed data. Thank you. If you have any questions, feel free to contact me at Baihan Lin at columbia.edu and check out our extended version recently impressed in algorithms. Thank you.